In this video, we're going to continue on talking about what happens in the brain. We're going to continue on talking about those biochemical steps needed for LTP, something that we touched on and started to discuss in the previous video, and now we're going to go into some more detail. Now, of course, LTP is theoretical. We can measure it in neurons, in petri dishes, in animals. However, is it really how we take what we've learned and form a memory trace? That is debatable, and at least some people would say maybe it's not the only way, but I think it's something important to cover in an intro to psychobiology class. So the first thing that happens in order for LTP to occur is repeated excitation of AMPA receptors. So I couldn't find a proper diagram, so I'm just going to try drawing this in here, just like I would on the board. And here we have our postsynaptic neuron, right? This is postsynaptic. And we have an AMPA receptor in here. And so what happens when your presynaptic neuron with its neurotransmitter releases that neurotransmitter into the postsynaptic cleft, it binds that AMPA receptor and in comes sodium. Well, one stimulation or weak stimulation presynaptically isn't going to be enough. You have to have enough sodium to depolarize the cell a lot. So it's repeated glutamate excitation of the AMPA receptors depolarizes the membrane by allowing a lot more sodium into that postsynaptic cell. So now remember, when we just have weak stimulation of AMPA receptors, that glutamate is still going to bind the receptor. This is the NMDA receptor now. But the NMDA receptor can't respond because of this magnesium block. When we have then repeated stimulation, we get a greater depolarization. We get lots of sodium depolarizing the postsynaptic cell, and that magnesium block is removed. So depolarization from repeated stimulation removes the magnesium block. That allows glutamate to then actually excite the NMDA receptor and open a channel for calcium ions to enter. So again, once we have that repeated stimulation, this magnesium block will be removed. It opens the channel, and now calcium can flow in. When calcium enters, it activates an enzyme called CAM kinase 2, or calmodulin kinase 2. You don't need to remember the whole name. CAM kinase 2 is just fine. Just understand that it's an enzyme. And there's a few other steps we're kind of skipping over. This enzyme then is activated, and through a series of events, it causes Krebs to be released and regulates the expression of genes. And so that's what you see here in this diagram. We're talking about a ligand-gated receptor. So this is the NMDA receptor. It's opened by glutamate stimulating it and depolarization of the postsynaptic cell by the AMPA receptors. Calcium enters, and it activates this enzyme CAM kinase 2. Krebs is also involved, but basically it goes in here and it can alter gene expression, cause protein phosphorylation, a whole lot of downstream effects that ultimately cause the changes postsynaptically and presynaptically that result in long-term potentiation. This regulation of genes and the response of CAM kinase 2 to the calcium is magnified by BDNF. And BDNF stands for brain-derived neurotrophic factor. And I mention it here because we will come back and we'll see it in a few other different situations later on in the semester. So it's important to understand that BDNF seems to play a role in plasticity and, again, magnifying that LTP, that long-term potentiation. So it's important to understand that CAM kinase 2, Krebs, and BDNF are all critical for this long-term potentiation. So in terms of postsynaptic changes with LTP, what happens postsynaptically? So let's say we have here, that's That's our postsynaptic neuron. So we're going to have more AMPA receptors that are going to be built or moved and put into this postsynaptic neuron. That's because that CAM kinase 2 activation allowed changes in gene transcription, which allowed more of these AMPA receptors to be built or moved. We also see an increase in dendritic branching. That increase in dendritic branching results in more synapses with the same neuron. AMPA receptors also become more responsive, and more NMDA receptors are inserted. So I can't make it a different color, but here would be more NMDA receptors being made, again because of that CAM kinase 2 activation. And these receptors becoming more responsive partly has to do with phosphorylation, which you saw also is something that CAM kinase 2 can be responsible for. So overall, we have increased responsiveness of the postsynaptic neuron. So this is that diagram from earlier showing you this is our normal response. We have stimulation. Once there's enough stimulation, sodium and calcium will enter. Well, after that happens and calcium activates CAM kinase 2, now we have more receptors, more AMPA and NMDA receptors inserted in the membrane. That allows this postsynaptic cell to have increased responsiveness. We also have some changes presynaptically. So there's a retrograde transmitter that can travel back to the presynaptic cell, causing the following changes. One is a decrease in the action potential threshold. So we talked about that threshold and said how most of the time every neuron has the same threshold for the duration. Well, once something has changed in the brain, once we've learned something and we're storing it as a memory trace because we're encoding it or we're consolidating it, that could potentially alter the threshold so that next time that cell will respond to the same stimulation more easily. Also, an increase in neurotransmitter release. That was also shown in that previous diagram on the last slide, where you could see that when the cell is potentiated, more neurotransmitters being released. There can also be expansion of the axons, so those axon terminals can sprout and change shape. It can go from one synapse into two, which is called bifurcation. And so that again increases the surface area of the synapse, allowing for more places for receptors to be inserted, more places for neurotransmitter to be released. And that leads to the last one, is transmitter release from additional sites. Again, overall, the idea is that we have increased activity in the postsynaptic neuron. And this is just showing you an example of how you could potentially see changes. The three-dimensional idea here is presynaptically, postsynaptically, and now your synapse is split into two, which again is called bifurcation. So is LTP important for all learning and storage of memories? No. As time passes, it appears that memory becomes more dependent on the cerebral cortex. And we've talked about that, how it seems like that those memories later on that don't have that context, that are a little bit more just the gist, seem to be stored in that cerebral cortex. And this is just an example kind of showing you that again kind of tying back when we started the beginning of this chapter to the end. We have sensory information, we have that short-term memory, which is occurring in the hippocampus. If we consolidate it, it gets into long-term memory in the hippocampus, which is what we're talking about now with those changes in the brain. But over time, it seems to go to the cortex. Now, could understanding the mechanisms of changes that enhance or impair LTP lead to drugs that block or improve memory? Yeah, but we're not really there yet. We know that caffeine and Ritalin enhance learning by enhancing arousal, increasing arousal, and we'll talk about that in one of the later chapters. So overall, the best way to improve your memory is to study better to begin with. Give yourself more cues to recall that information. Give yourself more time to try to remember that information.